Roughly 60% of applied fertilizers remain unused, directly contributing to water pollution and soil acidification. Additionally, long-term use of commercial fertilizers degrades soil health, leading to a decline in agricultural land.
Hello, I'm Thomas Nolan.
I'm Jaden Nguyen, and we're sophomores attending Westminster High School.
And I'm Tian Dao, a sophomore attending Garden Grove High School. Our project is titled The Effect of Arbuscular Mycorrhizal Fungal Inoculum Concentration on Growth of Lactuca sativa, and it aims to answer, what are the effects of different concentrations of arbuscular mycorrhizal fungal inoculum on Lactuca sativa plant growth?
Research shows that arbuscular mycorrhizal fungi form relationships with plant roots, acting as an extension of the root system, enhancing nutrient and water uptake, piquing our interest. Additionally, we wanted to test lettuce as AMF compatible and a significant agricultural crop. We hypothesized that the Lactuca sativa with higher arbuscular mycorrhizal fungal concentration will experience higher plant height because the arbuscular mycorrhizal fungi forms symbiotic relationships with plant roots to increase absorption of nutrients and enhance stress tolerance. Key materials we utilized was butter lettuce seeds, cherry bell radish seeds, Bliss arbuscular mycorrhizal fungal inoculum, 10 centimeter diameter pots, 100 milliliter beakers, seed starting soil, filtered tap water, Carolina Biological Supply Company with plant light box, along with a ruler for weekly measurement and a laptop to collect data.
In our 6-week span experiment, we grew 32 pots of AMF-resistant lettuce and 32 pots of AMF-resistant radish to validate that AMF is a driving factor behind differences in plant height, with 3 seeds in each box. We then added different concentrations of AMF inoculum to each treatment group, depending on soil volume: 0%, 0.5%, 1%, and 5%. These groups will continue to grow for a span of 6 weeks in a plant light box to regulate conditions where we will measure every week, collecting averages per pot and averages per treatment group.
After the experimental period, we found that the experimental groups with the highest AMF inoculum per volume of soil had shorter mean heights than those in the control group and in groups with lower AMF inoculum. The line graph shows that the increasing amounts of AMF inoculum led to decreased growth as the trend of the control group, represented by the 0%, having the highest mean height compared to the experimental groups, represented by the 0.5%, 1%, and 5% continued through the entire experimental period. At the same time, the bar graph of final height shows the same. The control with the highest mean height and with increased AMF concentration, the mean height is shorter. Additionally, we determined the statistically significant difference of our data using a one-way ANOVA test, which showed that our p-values were less than the arbitrary value of 0.05. This shows that the difference between the experimental groups and the control were significant enough to rule out that the chances for the shorter mean heights were completely within our experimental variability range. We also performed an individual t-test to analyze which groups showed the statistically significant difference and which groups had the most difference, and which led us to see that all groups were significantly different, with the control and the 5% having the most significant difference.
The scientific explanation for our data is that even though AMF typically enhances nutrient uptake, symbiosis involves carbon exchange costs to the host plant, which can often surpass the healthy limit of the plant. For the lettuce's mean height to shorten, the carbon costs outweigh nutrient benefit, which reduce energy available for growth and photosynthesis.
Overall, after our data collection period, we concluded that our arbuscular mycorrhizal fungal inoculum does not increase Lactuca sativa growth at these concentrations, indicating that our alternative hypothesis could not be entirely rejected and that further research at lower concentrations is needed. On the other hand, our data support the null hypothesis that our arbuscular mycorrhizal fungal inoculum can decrease the growth/height of Lactuca sativa. Our experiment helps identify the limits of AMF in assessing its positive effects on plant growth. However, we acknowledge that the results were small relative to within group variability, as evidenced by overlapping error bars. Additionally, we acknowledge that our experiment could have been due to our errors: frequent pot transport, accidental leaf destruction, and inconsistent soil nutrient levels. Therefore, while increasing AMF concentrations decreases Lactuca sativa plant height, we cannot conclude that this is always the case.